Hi, today I'm going to tell you how cancer gets so deadly. It can evade the immune system by hiding in plain sight.
The hypothesis for this is called immunoediting.
The first phase of immunoediting is called elimination. Elimination occurs before the cancer is clinically detectable. We all contain precancerous growths, or tumor cells that the immune system is initially effective against. This stage is also known as immunosurveillance. During immunosurveillance, many things can occur that cause an immune response. First, tumor cells that have a higher amount of activating ligand on their surface can bind to immune cells called natural killer cells and activate them. Cells can also express less of the MHC1 protein, which usually inhibits the NK cells. With less of this protein, the tumor can bind to the NK cell and activate it. When activated, NK cells take on many functions. They can directly kill a tumor cell by releasing proteins such as perforin and granzymes. The perforin creates an impermanent pore in the cell membrane, which allows for the granzymes to diffuse into the cell and trigger apoptosis. Lastly, these NK cells release interferon gamma, which trigger apoptosis of tumor cells and cause the maturation of dendritic cells. How do dendritic cells work? They engulf tumor-associated antigens that are left in the tumor microenvironment after a tumor cell lysis. They then present the antigens to CD8 cytotoxic T cells. These activated T cells release cytokines that trigger apoptosis of the tumor cell as well. The next stage is equilibrium. During equilibrium, there's a balance between the tumor cells that had escaped elimination and are continuing to grow, and the immune system simultaneously detecting and removing tumor cells. This is called immune dormancy, and it doesn't allow for metastasis. The tumor is also kept dormant through angiogenic dormancy, which is when tumors cannot grow due to lack of blood supply, and cellular dormancy, where the tumor cells are in cell cycle arrest and are not in a proper microenvironment to begin proliferation again. Furthermore, since only tumor cells with high immunogenicity are killed by the immune system, the cells that are resistant to immune effector cells are selected for and accumulate in this stage. This leads to escape. During escape, the resistant cells are able to proliferate and the tumor becomes clinically observable. There are many mechanisms for this evasion of the immune system. First, in the tumor microenvironment, large amounts of TGF-beta is produced. This protein stops T-cell proliferation and also helps induce the differentiation of non-T regulatory cells into T regulatory cells. Tregs, as they are called, suppress the immune system by inhibiting cells like cytotoxic T cells. If these effector cells cannot release cytokines, then the tumor cells cannot be destroyed. Another escape mechanism is antigenic modulation, which is when the surface antigens endocytose into the tumor cells or sheds and leaves the tumor cells completely. This prevents detection from the immune system as well.
Using this information, scientists are trying out immunotherapy so cancer cells no longer can hide.